Okay, today I'm going to start. We're now going to be talking about B cell receptors, immunoglobulin molecules, and how those are generated. So we'll start again with what questions that we want to address in this particular lecture. And the first question is, there are so many pathogens out there. How could you possibly make antibody to every possible pathogen? So again, this is a question in terms of how could you have such an incredibly diverse immune response? How do you avoid making autoantibodies? So how is there a specificity that only antigens are the ones that are in pathogens are the ones the antibodies are generated against? And how do you rapidly increase the amount of antibody? How do you mobilize an antibody response? Again, it doesn't do you any good to make great antibodies after the pathogen is basically replicated to such a high level that you're pretty much morbid and those antibodies won't be be effective in terms of protecting you from the infection. So you need a rapid mobilization of a response. And how do you switch from making IgM to IgG? And again, as I'll discuss when I talk about the different isotypes of immunoglobulin, IgM is the first antibody that you make, but it's not a very effective antibody. It's large, it has low affinity. The gold standard for antibodies is IgG. So how do you make this transition from making IgM to making IgG? And how do you increase the affinity of the antibody? Now, I kind of have to apologize to you because I lied a little bit, because when I talked about the fact that one cell, one antigen specificity, and that's the way it stays, it turns out that's true for T cell receptors. T cell receptors don't change, but antibody molecules actually undergo a large level of mutation to become higher and higher affinity. And in fact, it utilizes mechanisms that are actually similar to those that HIV utilizes in terms of mutation. So the same way that HIV mutates to escape the immune response, the antibody molecule has the capacity to mutate to become better and higher affinity antibodies to fight infection more effectively. So again, and you have to think that the genetic rearrangement is completely antigen independent, so how could you possibly know what the highest affinity sequence that you can make is before you've seen the antigen? And what this affinity maturation allows you to do is make even better antibodies. So how does that happen? And how do we generate memory? Clearly, once you've gone to all the work of making a IgG antibody that's incredibly high affinity, it seems it would be foolish to basically just throw out those blueprints and then wait till you get infected a second time and start from scratch. It makes sense that you have a system that enables you to have residual cells that have making these great antibodies that you could then call upon to expand the next time you're infected. So how does this process occur? So just to kind of give an overview again of antibodies, the process of antibody production is, in this case, you have a B cell. It is expressing antibodies. I'll discuss in detail how these are generated that recognize epitopes on the surface, in this case of a bacteria. This bacteria gets internalized, will get digested into the constituent peptides. The peptides are presented, and what MHC molecule class is presenting this? Class 2, very good. Because it's an endogenous from outside. It's presenting to a helper T cell, because CD4 class 2, that T cell receptor recognizes the peptide, and this helper T cell is now going to provide help. In order to do this, we'll discuss in a little bit of detail later on, there's also interaction with co-stimulatory molecules, so other molecules that interact on the surface, and the helper T cell makes cytokines. We'll discuss that in a little bit more detail, and this drives the B cell both to make antibodies as well as to class switch. The B cell differentiates into a plasma cell. The plasma cell is basically an immunoglobulin factory, makes large amounts of antibody, and these antibodies have very, very diverse activities. So some of these antibodies could neutralize infection. So by binding to surface areas on the bacteria, it prevents it from sticking. If the bacteria can't stick, it can't infect. Also, it functions to opsonize. So by binding to the bacteria, the antibody now is able to be taken up by receptors on the surface of macrophages called FC receptors. I'm also going to discuss in greater detail that allows macrophages to be more effective in phagocytosing bacteria. And in addition, and this I apologize for you, there's no lecture on complement in this short course, but complement also plays a role in terms of being recruited by antibodies to allow killing of bacteria. Now, if we look at the primary, secondary, tertiary response again, what we see now in this a little bit more complicated figure than the one I've showed you before, we're not only looking at volume of antibody production, we're also looking at what isotype is being produced. And what you can see is that early on, the antibody that's being produced is shown in purple and green is IgM, but as time goes on, the antibody that's being produced is the yellow antibody, IgG. So over time, you have switching from the initial antibody, IgG, to IgM. That's the top panel. But what's also interesting is in the bottom panel, the y-axis represents affinity. And what you can see is that the affinity early on is extremely low, because IgM has low affinity, and even when you're making IgG, the affinity is also low, but again, as time goes on, the affinity of the IgG dramatically increases, and I'll discuss later, the reason for that increased affinity is because of the somatic mutation that's occurring in the immunoglobulin gene. Oh, I'll use this then. Okay, so, well, what does the antibody molecule look like? The antibody molecule, you always see these cartoons, and when I first saw them, I always found it very difficult to figure out what exactly it means. You kind of like just nod your head and try to look smart, but it just looks like a bunch of colors and ribbons to you. So you have the more simple cartoons, and then when you look at the more simple cartoons, you think that's what it looks like in real life, and you're always surprised that it doesn't. But again, this is the simple one that I've shown you before, showing the antibody binding region, hypervariable region. In this other, little bit more realistic view, but still a cartoon, you could actually see that there's a hinge that allows these two regions in the heavy chain and the light chain to come together to actually combine to where it's binding the antigen. And again, in this ribbon diagram, it also looks like it's apparently the red is the light chain, the yellow is the heavy chain, and again, they come together. So think of it coming together like these two hands. So even though they're two separate chains, this one is specific, this one is specific, and now when they come together, the combination of the two that's what gives you the specificity. And again, as I mentioned yesterday, if you change the heavy chain or the light chain alone, you can now, by combining them with different heavy chains and light chains, dramatically increase your specificity. Okay? So this is, again, re-showing this clonal selection of antigen-specific lymphocytes, again, to reinforce the idea that you have this pluripotent cell that undergoes genetic rearrangements of the immunoglobulin gene. I'll discuss that in greater detail. But this gives rise to hundreds to millions of cells that all recognize different antigens because the antibody molecules that they have different specificities. And after you remove self-reactive B cells, I'll discuss in a later lecture how that occurs, you have a pool of these mature B cells, immunoglobulin molecule expressed on the surface. Initially, it's going to be expressing only IgM on the surface, and only when that particular B cell comes in contact with an antigen will it undergo expansion. You get expansion of millions of copies of that particular B cell, and then you clear the infection, and you retain many of those cells as memory cells. So if we now look at the genes, oh, thanks. If we now look at the genes, no, can't win. Okay, so can you hear me now? Yeah. Okay. So now if we look at the structure, so here you have the light chain, here you have the heavy chain. It's color-coded for you. So the variable region of the heavy chain is blue. The variable region of the light chain is yellow. And now let's look at the sequence of large numbers of immunoglobulin molecules and ask the question, where is the variability? And by doing the sequencing, what one sees is you see 3 hotspots within the variable region. So even within the variable region, there are areas that we call hypervariable. They have even more variability. And these 3 regions basically comprise different sequences, hotspots in the gene of the immunoglobulin molecule. Again, I'll show you what's the molecular basis for these hotspots. And this turns out to be where the immunoglobulin binds antigen. Because what happens here is that even though these hotspots look like they're at different locations in the gene, which they actually are, but now when the protein is made, the protein now gets folded, and the folded protein actually brings these 3 regions all together. So this region is the HV3, which is going to be over here. This region is the HV2, which is here, and this is the HV1, which is here. But now these hypervariable regions are actually brought adjacent to each other, and that's exactly what sees the antigen. So in fact, this hypervariable region is the most important part of the antibody molecule, because this is where all the capacity to recognize multiple different antigens comes from this hypervariable, these 3 hotspots of hypervariable region. And this is where there's the most sequence variation from antibody to antibody. And so now, how is this generated? And what we can basically demonstrate is that we have a multiple gene segments. So if you remember the T-cell receptor, we had a large number of V genes. We had a J region, and for the alpha chain, for the T-cell receptor, we had a V region, a D region, and a J region for the heavy chain, for the T-cell receptor, it was the beta chain. But now you have multiple V's, multiple D's, and multiple J's. You pick one V, one D, and one J, and then combination of whichever ones you pick, like a combination lock, different numbers, that gives you a high level of variability. And what's going to turn out to be the case is that the joining regions of the V, D, and the J, that's where the hotspots are. So in addition to having different V's, but this is where the high level of diversity gets generated. And you can imagine, again, as we said, the combination of the specificity of the kappa chain plus the heavy chain, that's what recognizes antigen, because, again, those 2 pieces come together to actually bind to the antigen. And this is basically what the immunoglobulin gene is going to look like. You basically have large numbers of V regions. So in this case, it's showing you 30 V regions, a large number of J regions, and basically for the light chains you have to have 1 V and 1 J come together. Even though these are the same color, the sequence of this is very different from this and very different from this. So don't get fooled by the same color. They're very, very different sequences, and therefore taking 1 V and 1 J is going to make one sequence protein. Taking a different V and the same J is going to give you a different protein, and so forth and so on. This is what allows you to get a multiple high level of variability. And again, if you want to think about it simplistically, it's almost like thinking of a train. And that train has 1,000 cars. Each one of those cars is a different color. And by taking different trains and putting them together, you can get multiple different combinations. Well, how does this happen? How do you get these different V chains to be approximated towards the J chain? And this occurs through a process called recombination. And if you think in terms of the train analogy, you know that trains are constantly being connected and unconnected and then connected again because they have unique coupling sequences at the end of the train. And the same thing occurs at a genetic level. Each one of these variable genes has switch sequences upstream and downstream of it that allows it to readily be coupled and be recombined. So in this particular case, what you're seeing is that you have the V region, V region, V region, and they're basically being looped out because switch sequences here are getting realigned, and then this basically gets cut out, and now this V region and this J region come together the same way you could take trains, uncouple them, pop them out, recouple the trains back together again, and you have a different sequence of trains. This is one molecular looping approach. This is a different looping approach, but the idea is exactly the same. You loop out the genetic information, so it's basically no longer there, but you now have the J next to different J's next to different V's, depending upon how much of the sequence you loop out. Is that clear? And if we do the math and ask the question how many different genes that we have, again, the kappa and gamma chains have 30 variable, heavy chain is 40. The D chains, 25 for the heavy chain. Again, there's no D chains in the light chain. And the J is actually also 6. Now, does that seem like a large number to you? Not at all. So if you look at that number, you would basically say there's no way you're going to get anywhere near the high level of diversity that you're going to need for antibodies. So what that's telling you is there must be a different mechanism there that's allowing you to get that high level of diversity. So we'll go into it to discuss exactly what that mechanism is. So again, this is just to show you exactly what those switch regions look like. Again, this is like the coupling sequences in a train, that there's this recombination signal sequence called RSS. On one side it's 23 bases, on another it's 12. These are flanking all the variable regions, these flank all the J regions, and they could recombine with each other. And therefore, again, this is the mechanism by which the V's and the J's can couple with each other and loop out. But again, how can you get even more diversity in the process? And it turns out that the mechanism by which this occurs is by introduction of random nucleotides in the joints between the gene segments during immunoglobulin rearrangement. So not only are you dealing with what you have, pre-existing genes, but now you're introducing new nucleotides randomly that can unbelievably amplify the level of diversity. And this is your recombination sequences. They're brought together. They make these hairpins at the coding turns, at the coding regions. But what ends up happening is a gene that introduces, it opens up now the sequence. But before you combine the two of them, you need to get a portable mic, because that's what you need, or a megaphone. But there's an important enzyme called TDT, and what TDT does is it just randomly introduces new nucleotides, and these new nucleotides can lengthen this area, and now ultimately, hopefully, you get the right pairing region, get rid of the unpaired nucleotides, and now you seal the DNA, fill it in, and now you have new nucleotides introduced that hadn't been there previously. Is that clear? Well, two things are really critical about this process. The first point is that, well, now you can introduce new amino acids. So if this encodes an arginine and it hadn't been there before, now you have a highly charged region that could recognize a different antigen. That's not anywhere near the cool aspect of this process, because you know about reading frames, right? So normally you have triplets. You go 3 nucleotides, amino acid, 3 nucleotides, amino acids, 3 nucleotides, amino acid, right? That's pretty straightforward. What happens when you add a single nucleotide into a DNA sequence? What happens to your triplets? What happens? They completely change, because one amino acid completely throws the entire triplets off. So not only you're not just changing one amino acid, you're changing all the rest of the amino acids in that local area. So you generate tremendous diversity. If you put 2 nucleotides in there, you throw off another reading frame. So in essence, this process dramatically increases the variability of the amino acids that are being expressed, and again, now dramatically increases the diversity of antigens that can be recognized by those antibodies. So this process, both of putting new amino acid capacity by putting a triplet in or throwing off the reading frame, dramatically increases specificity. And guess what? This is, again, one of those hotspot regions that have hypervariable sequences that are very critical in recognizing antigen. And now, if you start adding up all these different factors and do the math and ask the question, where is the diversity coming from? It turns out that the number of V gene pairs with D regions, et cetera, gives you this level of diversity in the T cell receptor immunoglobulin gene by having junctional diversity where the junctions don't exactly match. When you add nucleotides, you can see that dramatically increases it. You multiply these 2 out, and again, for immunoglobulin molecules, you come up with 5 times 10 to the 13th possibilities, and for the T cell receptor, you come out with 10 to the 18th possibilities. So that's a large amount of variation. A simple question may be, well, why does the T cell receptor have a lot more variability than the antibody molecule? So if you look at the numbers, what you clearly see is there's more variable regions in the T cell receptor, and particularly a lot more J regions in the T cell receptor. There are 61 J regions in the T cell receptor. Why is that? Why should the T cell receptor have a lot more variability than the antibody molecule? Hold that question, and we'll come to it when we start discussing somatic mutation. Okay, any questions? Okay, so now the antibody molecule has to do multiple things. One of the important things it does is it neutralizes viral infections. Well, in order to neutralize viral infection, it's basically the virus, for example, if you think about HIV, the receptor has a certain level of affinity for whatever the cellular receptor is. So for HIV, it would be GP120, CD4, CCR5. Well, if the antibody has less affinity for the viral molecule than the cellular receptor does, the antibody won't be very effective. Right? You can't compete if you have less affinity. So therefore, it's incredibly important for antibody to be usable as a neutralizing molecule that it has to have a higher affinity than the virus has for whatever its natural receptor is. And in order to get that high level of affinity, what the antibody molecule does is undergo a process called somatic mutation. And what's illustrated here is this is the genetic sequence of the heavy chain V region. This is the genetic sequence of the light region. And what you're seeing is during an immune response, day 7 of the initial response, you really don't see a lot of changes. Mutations are indicated by these little, whatever these are. How would you describe this symbol?
Pins.
Huh?
Pins.
Pins, okay, like little thumb push pins, round push pins here. Not a lot. Now, what happens is you look at 14 days, you start seeing a lot more. Then you have the secondary response. Now, by the time you're to the tertiary response, you could see there's a tremendous increase in the number of mutations. Every one of those little push pins, and this is from a single B cell, has more and more mutations. And where are those mutations localized? What area of the immunoglobulin region? The hypervariable region, which is indicated the CDR1, 2, and 3 by this shaded purplish region. So what's happening is that if you just take a B cell, follow it over time, sequence the immunoglobulin molecule over time, you see it's spontaneously developing these mutations that hadn't been there before. They're not in the germline. They're new, and what this is doing is changing the antibody recognition capacity, and presumably it's going to turn out it's going to make an antibody that has higher and a higher affinity, because the antibody is actually undergoing almost a Darwinian evolution, because multiple mutations are randomly being generated. Some of those mutations don't actually reduce affinity. If it reduces affinity, the antibody molecule can't compete for antigen, the B cell doesn't undergo proliferation and kind of dies off. The mutations that result in higher affinity compete better for antigen, and those are preferentially expanded. So over time now, you've selected for mutations that have even higher and higher affinity, and that's how you're generating these high affinity antibodies that you need for effective neutralization. Is that clear? Any questions? And this is just to illustrate anatomically how this happens. You start out with a pluripotent stem cell. The pluripotent stem cell, immature B cell, mature B cell, it sees antigen for the first time, gets T cell help, and now is triggered to undergo its first round of proliferation. When it's proliferating, it migrates into this dark zone, and it's called a centroblast. And this is a very critical time in terms of its differentiation, because as I'll discuss in a few minutes, an enzyme called AID plays a critical role in the development of these mutations. It undergoes random mutations, and now it comes in contact with antigen being presented to it by the follicular dendritic cells, and then there's a selection process so that B cells that have antibody molecules that have high affinity, they basically now are selected for. Some of them become plasma cells to make antibody. Some of them become memory B cells that will be around for the next infection. And these have affinities that are 1,000, 10,000, 100,000 times higher than the initial germline encoded immunoglobulin molecule. However, any story, there's always a sad part. You always have winners, but there's also losers. And you have B cells that unfortunately, they mutated in the wrong direction. Their antibody molecules had less affinity, and those B cells really are not going to be functionally of any value to you, and therefore they undergo apoptotic or anergic B cell death. So kind of like the same way Darwinian evolution can be very cruel, so too mutation in the germinal center can also be cruel, but it results in the selection of these very, very high affinity producing, IgG producing B cells. Okay, any questions? Yeah.
Just 3 slides earlier when there was a number of permutations that could occur. It was -5 times 10 to the 13th.
Yes.
Except the bottommost number. Why is it negative?
No, this is actually approximate. It's one of it's a squiggle. Oh.
Oh, okay, sorry.
This here is a
Right.
squiggle.
Yeah.
But and thank you for actually making me go back to this slide, because now I had asked the question, why do you have so many more possibilities in the T cell receptor than the immunoglobulin molecule? Isn't that bad for antibody production? And now you appreciate that it's not as critical, because the antibodies have the ability to undergo somatic mutation. So even though the germline you have 5 times 10 to the 13th, because you have the added capacity of undergoing these somatic mutations in the hotspot regions of those hypervariable areas, you can now dramatically increase your ability to recognize antigen many fold to even have, I mean, again, the numbers are ridiculously high, much higher than the T cell receptor. In contrast, as I mentioned yesterday, the T cell receptor does not undergo somatic mutation. Whatever rearrangement it underwent initially, it stays with that forever. So that's probably why you need to have a higher starting point in terms of possibilities, whereas the B cell, because it has the capacity for undergoing somatic mutation, can dramatically increase many fold beyond this baseline germline encoded variability. Okay, is that clear? Point clear? Okay, well, how does this happen? How are these mutations introduced? And this is through a molecule called, an enzyme called cytidine deaminase. And if people are familiar with HIV, where you have ApoBec3G, which interacts with VIF and causes mutations that can actually prevent HIV infectivity, AID actually utilizes a very similar approach. It's a cytidine deaminase. It takes the amine off the cytidines, changes them into a uracil, and that uracil will ultimately get translated into a thymidine. And it basically is associated with different RNA transcription enzymes that allows the introduction of several mutations. This is just a picture of what the enzyme looks like. And from a mechanistic point of view, what happens is that you have cytidines that are present in the gene. AID comes, deaminates it, and converts the cytosine to a uracil. And there are multiple different mechanisms that play a role. For example, one enzyme called uracil DNA glycosylase, or UNG, sees that there's a mismatch because G and U don't match. And again, there are anal compulsive enzymes. They're continuously checking to make sure that all of the nucleotides match. It's like, you know, anyone ever puts on a pair of one black sock and one blue sock in the morning, and it's dark, you don't notice it, and then all of a sudden you look around, you walk in with a blue sock and a black sock, it looks really pretty nasty. And if you're married, your spouse points it out to you and then says you better go back and change it. You know, these enzymes do exactly the same thing. They're continuously making sure that the nucleotides match. If something doesn't match, it basically pops it out and forms a nick in it, and now you have a nick here that theoretically should be filled in with the appropriate cytidine, but sometimes that doesn't happen. Again, this is one of the mechanisms by which the somatic mutation gets generated. And these are the different possible things that can happen. Well, one possibility is that you don't notice it. And if you don't notice it, the uracil is seen by DNA polymerase as a thymidine, and therefore when it replicates, instead of putting the appropriate guanidine over here, it says, oh, this is a T, what goes here is an A. So right now you've now made a mutation. You've gone from a C to G to a T to A. Well, why is that important? Because now that's encoding a completely different amino acid, and that now is giving a variability to the immunoglobulin molecule. In addition, there's a lot more complicated processes that go on that you excise that, and now you introduce randomly the different nucleotides. But what gives even higher level of variability is that somehow this nick, which is open, gets expanded, and now you have a large gap in your DNA sequence. Well, if you'd be using a polymerase that has a high fidelity, that wouldn't be a problem. You just put back the appropriate nucleotide. But let's say if you're like a bad speller, so you don't put the right things in right, there are enzymes that are very error-prone, and this particular error-prone polymerase is specifically recruited by PCNA and actually fills in the blanks, but it fills it in with the wrong nucleotides. So now it's another way of generating all these mutations, all started because the AID made this single mistake that now gets amplified into more and more mistakes. Now, if this would happen in a normal gene, we would be in big trouble because we'd be making proteins that don't work, we'd be making mutations in oncogenes and beginning cancer. It seems for some unknown reason that this particular process only occurs in the immunoglobulin gene. Immunoglobulin gene is the only gene where this high level of error is actually encouraged. But again, it only occurs in this narrow window of time during B cell proliferation when it's in the dark zone, and it doesn't occur after the B cells mature. And if you look at the level of mismatch repair, this is a very low level of mismatch repair, 10 to the minus 11. That is healthy. You're fine because you're having very, very high fidelity replication. However, as you increase the error rate, you can see bad things start happening. So when you're at an error rate of 10 to the minus 9, that's not compatible with life because your cells just aren't replicating appropriately. If you have, in this region, you start getting cancer because you're basically mutating oncogenes. But it turns out that when you have this level present in B cells, that's actually works out well because you actually have this focused, targeted mutations that are only occurring in the immunoglobulin molecule, but this dramatically increases the capacity to recognize with higher affinity antibodies. Now, after T-cell dependent activation, you undergo mutations and selection, and now the B cells that you generate, well, these particular ones are the losers. So these B cells, they mutated their antibody molecules, as I mentioned before, but these were lower affinity antibodies on their surface. Therefore, they can't compete effectively for antigen. The whole process is antigen driven. If you can't capture antigen to the B cell, that B cell will stop proliferating and undergoes apoptosis. The ones that have higher affinity antibody, those now are successful in competing, and they become the plasma cells that are making high affinity antibody, and those also become the memory B cell. Well, that explains why the second time that you see an infection, you have a rapid response, but a more effective response, because you go to these memory B cells, and these memory B cells are now making this high affinity antibody that you spent all this time selecting for. So you can mount a very rapid response with a high affinity antibody, and therefore clear the infection very rapidly. So in essence, the B cells is learning from its mistakes, utilizing the mistakes that actually work, and doing it effectively to allow you to have a more effective immune response. Is that clear? Any questions? I mean, it's really an amazing process that occurs. Now, I mean, one teleological question that you could ask is, well, why don't T cells have the same process of somatic hypermutation that B cells do? Why do B cells need it maybe and T cells don't? Any thoughts?
It might be that the intensity of the interaction that you need with B cells is far more important than the intensity of the interaction that you need with T cells. Because T cells are essentially secretory. Well, if you look at alpha T cells, they're just there to secrete.
Okay, so hold that thought. So you're basically starting out by saying that B cells need to have an immunoglobulin molecule that's very high affinity. T cell receptors don't have to have as high affinity. Okay, that's point one. Let's take that to the next step. Why is that? What is the effector molecule of the B cell?
The antibody.
It's the antibody. That antibody is soluble. It's floating around. Well, clearly, it has to have a very high binding affinity in order to be effective while it's going to bind to whatever it binds to. So that's the major role of the antibody molecule, is to bind to things. If the major role is to bind to things, you want it to bind as tightly as possible, right? What's the function of the T cell receptor? It's a switch to turn on the T cell. It doesn't have to compete for antigen free-floating, because the antigen-presenting cell brings it to it, shoves the peptide right in the face of the T cell receptor and says, here this thing is, do you recognize it? It doesn't have to have a high affinity. As long as it recognizes it, then it transduces the signal. So you don't have that need to bind with high affinity that the soluble antibody does. Is that clear? Okay. And a second explanation is actually that T cells are very, very dangerous if they recognize self-antigen. A question that you may want to posit to me is, well, doesn't this somatic mutation lead to the possible generation of autoimmune antibodies? Because let's say you make a mutation randomly, you've left the bone marrow, you've left your screening location, and now you're giving B cells a license to make any antibody they want with any specificity. Could you now make autoantibodies, right? And the answer is, what do you think? Yeah, you can, absolutely, and it happens. And in fact, that's one of the mechanisms by which autoimmunity is generated. However, it turns out that antibodies, they're just not as deadly as a cytotoxic T cell. If you have a cytotoxic T cell, as I'll discuss in a subsequent lecture, and now it's recognizing a self-antigen, it can now go on, proliferate, and make millions of copies of itself, and go on and kill millions and millions of cells, and have a very, very vigorous, powerful autoimmune response. So because of the fact that cytotoxic T cells are so deadly, you therefore have to be extra special careful that you don't become autoimmune, and therefore the immune system has made a decision that T cell receptor maturation stops in the thymus, Only those that are okay in the thymus, they leave and they never change. So those two reasons probably account for why antibody molecules undergo somatic mutation, while T cell receptor does not undergo somatic mutation. Okay, is that clear? Is that helpful? Okay, so now this is just to kind of end as a lead into the next lecture. So this is also just to review. So now I think you now have an understanding of what's going on in this figure, because the initial reaction that you have is IgM. It has low affinity because the IgM has not undergone any somatic mutation. The immunoglobulin gene it's expressing is the one that was randomly generated by VDJ recombination. That occurs in the nucleus in the complete absence of antigen. So it has no idea what the antigen looks like. It's just randomly generating them. So it's going to be low affinity. However, as time goes on, through multiple exposure to antigen, you undergo somatic mutation, you mutate the VDJ region, particularly those hot spots, and you're undergoing this evolutionary process selecting for higher and higher affinity immunoglobulin gene sequences, and now the affinity dramatically increases. And again, that's also, you have higher levels, higher affinity, much more effective antibody response, which is why by the time you've been infected the second or third time, those antibodies are so good that the pathogen really doesn't stand a chance, and you're able to eradicate it very, very quickly. Okay, is that clear? So now, how can we make antibody to every possible pathogen? And the answer is, you have all these VDJ sequences. You have diversity just by having these different sequences, even more diversity by virtue of having when they rearrange, the junctions don't exactly match. You introduce new nucleotides into those hotspots, and then you have the additional benefit of having somatic mutation to further increase diversity as well as specificity. How do we avoid making autoantibodies? We haven't answered that question yet, so we'll keep that on hold. We'll talk about that in a subsequent lecture. How do we rapidly increase the amount of antibody? Again, by having these memory B cells, they're available, and because they have high affinity, then even a small amount of antigen they rapidly grab onto and start getting activated very quickly when the infection initiates. How do we switch from making IgM to IgG? That we're going to be discussing in the next lecture. And the way we increase the affinity of the antibody is through affinity maturation and through introductions of mutations, AID, deaminate cytidine, makes it into a uracil, and that starts a whole process of repair that's specifically designed to be inefficient and to be full of mistakes in order to allow this diversity to be generated. And how do we generate memory? We're going to be discussing that in the next lecture. Again, thank you very much for your attention.